Okay, thank you. So my talk will be about genes, mutations, and interactions between mutants, and what can they tell us about the function of the genes in the cell. So there are many ways to understand the role of a gene in the cell, and one of them is to disrupt the gene by generating a mutation and look at what the effects of this mutation are. In yeast, luckily, this has been done systematically almost 10 years ago now, by creating a collection of mutants, each deleted for a single gene. And what people showed were using this is that only 20% of the genes were essential for survival, for viability. And this obviously doesn't mean that the remaining 80% are not important, but it does say that there is a huge amount of redundancy going on in the cell, meaning that at least 80%, the absence of 80% of the genes can be compensated by the presence of some other component in the system. And what it does say is that if we're interested in understanding how redundancy works and what genes compensate for each other and how they do that, we have to start introducing multiple mutations to the cell. And the double mutants is the first logical step towards that goal. So for example, if you imagine you have two parallel pathways that ensure the execution of a common function, the cell is expected to be able to tolerate the loss of a component from either one of the two pathways, but not from both. And when that happens, when two genes are deleted in two parallel pathways, the function cannot be completed anymore and the cell is lethal. So this phenomenon is called synthetic lethality, and it's a part of a much larger class of events called genetic interactions. The interesting part is that synthetic lethality and genetic interactions are not just important to uncover genes that buffer each other. They can also help you to understand which genes work together in the same pathway, protein complex, or any kind of functional module you can define. And the way it works is that you imagine that if genes A, B, and C work together as, for example, sequentially in a pathway, they will have exactly the same interaction partners with the parallel pathway. So what this says is that patterns of genetic interactions or profiles of genetic interactions can be used as a very powerful functional prediction tool to uncover novel components of known pathway or just define groups of genes that work together. The only condition in order to do this and on a decent scale is that you actually need a lot of information about double mutants. And again, luckily in yeast, it became possible several years ago with the development of the synthetic genetic array, which is basically just a high-throughput method for constructing double mutants in yeast. And it takes advantage of the yeast deletion collection on one side and of very specialized robotics on the other side. The way it works is very simple. You have a query strain that carries a mutation in your gene of interest, and you cross it to the entire yeast deletion collection. And then a number of robotic steps spin the cells from one selective media to another to generate an output array containing just the double mutant progeny of the strains you originally crossed. And the robots who do that look something like this. Stacks usually filled with plates that get moved around and pinned and stored for growth into incubators. And when these machines are at full capacity, we can normally do about 200 genome-wide genetic screens a month. But a typical SGA plate, an output SGA plate, usually looks like this. It contains about 300 double mutants, each present in quadruplicate, the 4 colonies that are right next to each other. And what we do is we take digital pictures of these plates and then process them through an image processing software that measures colony sizes in terms of pixels, and that's what we use as our quantitative measure of fitness for all our double mutants. And when we started doing this systematically, we very clearly realized that colony sizes, as they are affected by many experimental factors that have nothing to do with biology, and they're just the way the experiments are done. Particularly here, I don't know how much you can see, but these colonies on one side of the plate are consistently smaller than colonies on the other side. And this is, per se, is not unexpected, given this is a high-throughput method, but we were surprised to see to what extent it actually affects our measurement. And we spent quite some time, long time, trying to identify and understand this effect. And we were able to cut it down to 5 major factors. Unfortunately, I don't have time to go into detail to explain what kind of effects they are, how we address them, but I would like to highlight just one of them, what we call the batch effect, because we think this is one of the major contributors to the systematic variance of our colonies. And the batch effect manifests itself is in the fact that screens performed together on the same day by the same person on the same machine tend to have very similar genetic interaction profiles that obviously have nothing to do with the genes that were screened on that day. And that's obviously a big problem for us as trying to use genetic profiles as a functional predictor. But the bottom line is that after about 2 years trying to address and deal with all these effects, we were able to obtain double mutant fitness information that was clean and unbiased and was now ready to be used to identify genetic interactions. So as we have a quantitative measure of genetic interactions, a quantitative measure of double mutant fitness spectrum, we needed obviously a quantitative model for measuring genetic interactions. And there is a little bit of debate about this in the field, how genetic interactions are measured, but most people would agree with this kind of definition. So you can imagine you have a wild-type strain that has a reference fitness of 1, and 2 single mutants that might have some fitness defects. So in this case, they might have a fitness of 0.7 and 0.5. So most people would agree that if the 2 genes have nothing to do with each other, if the reason they have fitness defect are completely unrelated and not biologically linked, then the double mutant is expected to have a fitness of 0.35, which is the product of the 2 singles. When you go measure the actual double mutant fitness and you observe that it's significantly lower than expected, then you call it a negative genetic interaction. And that's where synthetic lethality lies in this category. On the other hand, if the actual double mutant fitness is much higher than you would expect it, then it's a positive genetic interaction. One example of this type of interaction is suppression, where the double mutant is healthier than the sickest of the 2 singles. And what this model allows you to do is to define a score, which a lot of people refer to as epsilon for epistasis, but we call it just genetic interaction score, that is given you by the difference between the observed double mutant fitness and the expected double mutant fitness. If this score is negative, then you have a negative interaction, and vice versa. If it's positive, it's a positive genetic interaction. So we obviously wanted that our method works in the way we expect it to be, and we especially want to understand if all the correction normalizations we incorporated actually make a difference. And obviously, luckily, it does make a difference. So here I'm evaluating similarity of genetic interaction profiles measured by computing Pearson correlation between profiles. And I'm comparing genetic interactions measured with the entire method that we developed compared to the same method with all the normalization excluded. And as you can see, this makes a very big difference. This is a precision recall curve. And we've the gold standard we are comparing this to is a set of gene pairs that are co-annotated to the same gene ontology term. So it's a functional standard, gold standard. So after we saw this and many other evaluations that we ran, we convinced ourselves that our data is good, is clean, and we can now use it to ask a lot of interesting biological questions. And at this point, our data set was quite consistent. We had about 1,700 genome-wide screens with 1,700 queries crossed to about 4,000 non-essential gene deletions. This covered about 6 million total gene pairs, which is about 30% of all possible pairs combinations in yeast. And we generated 170,000 genetic interactions, positive and negative, which altogether represent about 3-fold increase over what was available up to that date. And the most important question we wanted to ask with this data is, can we use all these pairwise genetic interactions to draw a functional map of the cell, such that genes that work together would be located together on this map, and genes that were more independent from each other would be further apart. And we decided that we're going to use the same idea of genetic profiles to do that. So as a reminder, we know that functionally related genes are expected to have similar genetic interaction profiles. But now we had quantitative genetic interactions, so we visualized our problem as having a matrix of interactions, genes on one side and genes on the other, and each profile was composed of positive and negative genetic interactions. So what we can do here is compute pairwise Pearson correlation between every pair of rows and every pair of columns, and we can try to visualize it as a network. In this network, nodes will represent genes, and the edges will represent correlation of profiles. Edges can be weighted according to how high the Pearson correlation is for this, for that pair of genes. We can then apply a network layout that will try to reorganize the genes, basically a cluster network, such that tightly and highly correlated groups of genes will be located close to each other, and loosely correlated genes will be further apart. And when we applied this approach to our entire data set, we obtained something that looks like this. So here, once again, every node is a gene, and they are connected, every pair of genes is connected by an edge if their genetic interaction profile are more similar than a Pearson correlation of 0.2. And the network layout reorganized the nodes into a bunch of like 10 or 12 major big clusters. And one obvious question is, what are the genes in each cluster? And we went to look at functional enrichment in each of them, and we found that, for example, the cluster is hugely enriched for genes working in DNA replication and repair. These guys in this cluster are enriched for pretty much all genes involved in ISCB transport and secretion. And in fact, every single group of on the map was enriched for one major functional category in the cell. So this is interesting for a number of reasons. For the first time, we were able to put all the genes that were that we have so far relative to each on their functional relationship. Second of all, not only the clusters were the map, but also the relative position was informative in many cases. For example, DNA damage, DNA replication repair, and chromosome segregation were always, despite the fact that they constituted two completely different, they were always positioned next to each other, indicating that they were crosstalk. There is a lot of crosstalk going on between the two groups. They seem to be made for polarity and cell morphogenesis genes and cell wall related function. The other interesting aspect of this is that you can actually take this approach, apply it to a subset of genes, basically zooming in on parts of this network and getting more and more, a higher and higher resolution functional annotation about genes. So in this example, we're focusing on this area of the big map. We're extracting the genes from the and reapplying the layout, and now we again see genes clustering into groups and positioning themselves relative to each other. And go and see what they are. You notice that now you have again still clusters, now detailed. You can have individual pathways and even protein complexes, and you can start looking at how they are positioned relative to each other. Every black node on this map is an unknown gene. So right now, right from here, you can start making functional prediction about what you think that gene. And you can take this approach even one step further and set a subset of these genes. For example, I'll be focusing on these green nodes involved in biosynthesis and transport, and you can get an even higher resolution of what these guys do. So here, once again, I'm zooming in from this area to this area here to a higher resolution map, and in this case, I can start drawing direct interactions between them. So I'm not looking at correlations anymore. Now I have few genes enough for me to draw just the direct network. And what you see here is you have a couple of amino acid biosynthesis pathways that are synthetic lethal with each other and synthetic with this pathway that is involved in GAP1 sorting. GAP1 is an amino acid permease involved in making sure, the pathway is involved in making sure that this permease is properly localized onto the membrane. And it is responsible for ensuring that amino acids get transported from outside the cell into the cell. So it does make sense that amino acid biosynthesis, amino acid import are synthetic lethal because you need one of two processes to survive. What's interesting is that next to genes that are not involved in this pathway, we found 3 genes that were completely uncharacterized in relation to this process, but had exactly the same genetic interactions to known components. And unfortunately, I don't have time to show you the details, but we did do, our collaborators actually did very elegant experiments to show that actually the phenotype associated with the mutations of these 3 genes are very consistent with their potential role in the GAP1 sorting pathway. So, per se, this approach is not unexpected. So we knew interactions were informative. They have been studied for several years now. But what's unique about this approach is that I'm extracting this data from a data set that has about 60 data points. And I'm showing just one example out of the thousands that we found that are in the data waiting to be identified and experimentally confirmed. To facilitate this process of identification of potential novel biology, we drew, I think, 8 or 9 maps. See one I showed you that cover all the major clusters. And this is available for download from our supplementary material. And what this map also visually represents, it represents the common concept that nothing in the cell is isolated. Everything is connected to everything, and we have to keep it in mind when we do our analysis. Another way to look at this is to actually quantitatively measure what kind of interactions, how many interactions occur between different processes. So we did this with 17 broadly defined functional classes, and we measured how many genetic interactions, in this case negative genetic interactions, occur within and between these groups of genes. So from this heat map, the brighter the color means the higher the number of negative genetic interactions, and blue means lower number. So a couple of interesting observations from this heat map. First of all, among the brightest spots on this heat map is the diagonal spots, which represent interactions within processes. So this was known for quite some time because people understood for quite a long time that genetic interactions tend to occur between function-related genes. But what's interesting is that there are certain processes that are very underrepresented for genetic interactions. And there are several reasons why that could be the case. One of them could be that we're not screening these particular genes under the right conditions, or there is such a high level of redundancy that double mutant cannot uncover it. What's also interesting that some processes are actually super enriched for genetic interactions, both within and between different processes. Chromatin and transcription is one striking example. It tends to have interaction with pretty much everything, and that's consistent with the central role of gene regulation and the whole economy of the cell. And what's interesting is that this confirms a previous finding in completely different organisms. Here, chromatin and transcription genes were also found to be central to the network. What was surprising to us is that in addition to chromatin transcription, we found secretion-related processes also to be very central. And in this case, and this is not clear to us why that is, but it's very, very interesting, and we're going to follow up on that. We also wanted to know what are the properties of the individual genes that show a lot of exceptional number of genetic interactions compared to others. So in order to do this, we analyzed the relationship between genetic interaction degree and a number of physiological features that we thought were interested. And in particular, here we're measuring Pearson correlation about interaction degree and each of these different features. This most striking correlation we found is with single mutant fitness defect. The more severe the fitness defect of a gene, the higher the number of interactions it will have. So this is consistent in the fact that genetic interaction hubs are central to the system. And if you delete that gene, a lot of things can go wrong. Consistent with that, genetic interaction hubs tend to have a lot of Go annotations. That's what multifunctionality measures. It's the number of different Go annotation every gene has. They also tend to have a lot of different phenotypes as measured by the phenotypic capacitance. And what's interesting is that we're comparing here genetic interaction hubs, positive and negative, to PPI hubs, protein-protein interaction hubs. It was known also that protein-protein interaction hubs also play a very central role in the system. But here we're showing that, yeah, they have the same trend going in the same direction, but genetic interactions do so in a much more striking way. This was very surprising to us and very interesting. Protein-protein interactions hubs and genetic interactions have a lot of common features. For example, they tend to be highly expressed, highly conserved, and they evolve relatively slowly. So what we also can do is that take all these properties and use them to predict how many interactions the processes would have if our understanding of the system was correct. So in the previous slide, I show you the actual number of interactions between processes, and we found that if we use these properties to predict this, the interaction between processes, we get a correlation, very high correlation, which is around 0.8, which indicates that our understanding of how the system works is actually pretty close to how it probably works. And one important consequence of these findings is that organisms that don't have very easy genetics right now can be, in those organisms, genetic interaction hubs and all these properties can be used to predict genetic interaction hubs. So if we have a sequence of a particular genome, we can probably try to predict what kind of genetic system it will have. So one last point that, one last application that we used our genetic data for is to integrate it with chemical data. So it's a common concept that many drugs can mimic the effect of deleting the target of that of the drug. So in this case, you can imagine you can block this function by either deleting two genes that work in two parallel pathways, or by targeting one of them with a specific small molecule in the presence of a second mutation. In the same way, you can imagine that a drug's sensitivity profile, so the set of mutants the drug is sensitive to, so that are sensitive to the treatment with a particular drug, should be very close to the genetic interaction profile of the target of the drug. So the concept has been also around for a while, but now for the first time we had the data to actually test that. And we did this by taking our genetic interaction and mapping on the top of that all the drugs from a couple of published studies. Here, circular nodes are genes and triangular nodes are chemical compounds. And we placed every chemical compound next to the gene whose genetic interaction profile resembled the closest to the chemical profile of the drug. And obviously, many known drugs got placed exactly where you expect them to be. So hydroxyurea, known DNA replication inhibitor, was placed exactly in the middle of the DNA replication cluster. Same thing for nocotazole and tunicamycin and many other examples. But what obviously we are mostly interested in is unknown compounds. So it was one of our top candidates, this unknown compound named 0428, 0027, and it clustered right in the middle of glycosylation syndrome. So we went to see what is the gene that this drug profile correlates to, and it turns out is this ER1 gene that is an essential component of the oxidative protein folding pathway. And the ER1 genetic interaction profile had a significant number of hits in common with the compound chemical profile. And the compound is now, we suggested to be renamed to Erodoxin to reflect the fact that ER1 is the target of the gene. We did a number of, our collaborators actually did a number of experiments trying to confirm this, and one of them showed that the treatment of Erodoxin actually delays the oxidation of TRX1, which is a homolog of the ER1 real substrate. So altogether, in conclusion, our data showed that systematic genetic interaction screen can produce a map of the cell that has never been shown before, that shows functional relationship between genes, that genetic interaction hubs correlate with a number of fundamental genetic properties that we use to predict to make interesting predictions in higher organisms, and the genetic interaction network can be used as a base for chemical genomics analysis. And I would like to thank all the many people who were involved. This is a very big project. In particular, Michael Costanzo, who coordinated the experimental work of this project. Obviously, Charlie and Brenda for the overall support, and this incredible collaboration with Chad Myers at the University of Minnesota, who supervised most of the computational work for this project. And obviously, our funding and the fellowship. Thank you very much.
Yeah. You have time for questions? Or?
Yep.
Yeah, first, the interaction explain